We get experimental science. We're curious, non-judgmental.
Hey, everybody.
There he is. Dr. Guppy, how are you doing today? Thank you for coming back.
I'm ecstatic to be joining this audience again. Like, I had so much fun last time. I have to be here.
Awesome. Thank you so very much. And what time is your heart out, just so that I know, and I want to be respectful and we don't take you too far over.
This time I actually went ahead and made dinner already. So heart out is at 7 when dinner starts with my daughter. We're very regimented with my daughter, just to make sure that nothing goes overboard, or bedtime is sacrosanct in this household.
We're my little one too. I mean, she's much younger, but we're trying to get that timing down, that exactly when bedtime is. It's right after the bath. That's when we have a meal and we go to sleep, and always the same approach each time.
And I'll non-science real quick, as a dad, being on that is so crucial, right? Like, you think about how their brains are developing and how they're collecting new information. Everything is so new that the routine at least makes sure that there's that consistency. Because imagine if you were being faced with something new every second that you were alive. That would be crazy. We all crave some of that routine, so that not everything is overwhelming. So little dad to dad advice here. Just
appreciate you. So could you give us another intro on who you are again? I know we did last week, but just for anyone new coming in that didn't make the stream last week, if you can just give a quick intro.
Sure. My real name is Amitabha Gupta. Pronouns he, him, his. But everybody has called me Guppy since 4th grade back in India. I did not become Guppy when I came to America to make it easier for Americans. I've been Guppy for a long time, but that's the bonus. And I did my PhD at Columbia in DNA damage and repair, and I came to Fred Hutch to do my postdoctoral research to become, I wanted to go become a teacher at a small liberal arts school, but Fred Hutch convinced me to stay and teach not just students, but external audiences about the science that Fred Hutch is doing. So I've sort of created my own role and my own job to go out and really learn all the science that Fred Hutch does and find a way to bring it to everybody, and also find amazing science communicators like soon-to-be Dr. Elizabeth Bonner, who was absolutely phenomenal. I absolutely loved her talking about so many aspects of her research.
Yeah, that was really, really cool. And I love, again, looking at the slides and seeing, in real time, the differences in the nuclear architecture and the effect that would have on the cancer. I think, again, seeing that and, us wiggling around the scope, and we'll look through and see more of these structures after Dr. Guppy, but I think it'll be a really cool thing to be able to look around and actually see these differences, which is really exciting to be able to do that.
I'll just type in my real name into the chat, as Pragmatic Entropy is asking. And so I'll say this, blood cells are just so cool. There's, they all start out from that single stem cell, and they become every different type of blood cell in your body, each with different functions, each with different structures, each with different roles. And it's crazy that it all comes from one single source, and that just messing up with that original stem cell can make so many changes going forward. It's absolutely mind-boggling to me.
Yeah, and there's so many places where it can go wrong as well, and there's so many places where, as Elizabeth was telling us, they're trying to correct it too, and targeting the bad populations, which is, I think, it's encouraging, but also wow, there's a lot of potential places where things could go wrong as well in terms of where you're targeting.
Yeah, I mean, and like she also said, it's a miracle that we don't always get. The miracle that stuff actually works normally is there's so many finely tuned elements, from how the DNA is packaged and put in one space, how it's replicated effectively, and no errors are made. This is meters and meters of DNA that are being replicated every day, without error, at a really low frequency of error, how those are then converted into RNA, the right RNA for the right cell, and the right fragments are pulled out, and you don't end up with a wooden spoon in your cheese souffle, your whatever meal option she was talking about, and then how those proteins perform their functions, and then the cells end up dividing appropriately, and every cell ends up with the right number of. I mean, there's so many places that even just a slight switch can really have profound effects.
Yeah, and so some of the really cool things we were talking about last week when you were on were other avenues of research at the Hutch that were beyond what Pravrutha and Elizabeth were telling us about. And in chat, someone had questions about machine learning and how that might correspond to cancer discovery or cures. Could you tell us about if there's anything like that happening at the Hutch and a little bit about the science behind it?
Totally, and I'll actually sort of jump off of a little bit of what Elizabeth was talking about, right? Like, so in her lab, they're starting to do these like drug screenings, right? So you take FDA approved drugs and you test which works, which what doesn't. So a lot of these drugs inhibit a class of proteins called kinases, which are often sort of they they check whether things are working or not, and they add these modifications called phosphorylations. So they're like on switches in your body, and for a lot of cancer cells, those on switches are altered and permanently on. And so a large number of drugs target those kinases specifically, but it's never specific. It affects like a lot of random. It can affect multiple kinases because just the way that those kinase proteins are structured means that something that blocks one could block a couple of other ones along the way. And so Dr. Tarun Gujral at Fred Hutch has developed a machine learning algorithm where he's able to test a whole bunch of these kinase inhibitors, and based on how your cells respond to those, sort of narrow down exactly which kinase is being directly targeted
Oh,
based on
wow.
how you respond to a certain combination of drugs. And so he's taken this and applied it all the way across, and now he's doing some really great work in like orphan diseases or rare cancers, where basically he's partnering, as Elizabeth alluded to. The beauty of Fred Hutch is you have researchers and you have doctors like right across the hallway, and so doctors are getting like sending him tissues from tumor tissue from patients and healthy tissue, and he's making these slices and testing with a small subset of drugs, and running this machine learning algorithm to identify what is the which drugs work best, or which drug combinations work best for the tumor versus the healthy cell, and sort of then getting the treatment to the patients on sort of compassionate use. And these are already approved, so you don't have to worry about like safety or efficacy.
That's
So
amazing. And so
I know.
I, I'm just wondering at the logistics of that. How many? Oh, and Raven, thank you for the $10 dono. 1530. Thank you so very much, Raven. How does one like? How many do you have to test? Like, I'm just trying to visualize like what this experiment looks like, right? Like, is it how
Yeah.
many combinations do you have to test in order for the algorithm to know or be able to predict a quality treatment for the patient?
So I think right. So the big benefit of what he does is he takes the patient tumor and he slices the whole tumor. So we talked a little bit about this last time, but just to bring it up again, like solid tumors and certain other tumors, they don't just exist in a vacuum. They sort of exist with healthy cells around them, some nerve cells, maybe some immune cells, and they exist as sort of this architecture that is really critical, and they all work together. And so he he preserves that architecture in these tumor slices. And so I think with as few as like 50 to 100 drugs, he can actually predict combinations that would work best.
Gotcha.
Yeah.
That's really really amazing, and it could be personalized, like you were saying, every single patient. Do you know by chance the time scale? Like, how long does something like this take?
So I think something like this is probably running at yeah. So handmade art, you're right. They've created their own ML algorithm Uh, they actually created their own ML algorithm. Sorry, but they didn't. This is not like publicly available stuff, and so they. I think I heard once 10 weeks, but I could be very wrong. It could
Okay.
be less than that. But
Okay, cool.
once you figure out the right combination, you want to go into validation. You want to test and retest just to make sure you're giving somebody the um, right sort of match.
So it's not just for folks to get understand the pipeline of it too. It's not that you just drop a couple of drugs on, you get an output from the computer, and you go right to the patient. Right? There is a
Yeah.
couple of additional steps. Do you by chance know what some of those additional steps are to ensure it's safe for the patient?
So ideally, we already know that the drugs are safe, right?
Okay.
Like that's why he's using FDA approved drugs. They're safe and they're approved. So I think first, like the slices have to be made. They have to be brought. The tissue has to be taken out from the patient, brought to the lab, slices made, and started to grow
Mm-hmm.
in the lab. And once they start growing, then you can add the drug and see how long it takes before and then you have to give it a couple of days at least to make sure that like healthy ones growing, non-healthy ones not growing, and then you run the algorithm and then figure out what combination works, and then you apply it to you test out that plus like near neighbors, or like the algorithm may not predict exactly one combination, it may give you four or five, and you test those and sort of other close associates along with it as well.
Gotcha.
So
When you get this new combination, presumably that combo hasn't been tested in a person before. Do you need reapproval from the FDA for something like that? And how do you know that this new combo is safe? Like even like maybe individually that they're safe, but not in a combination.
So I think there's so much information out there that a lot of these have already been tried in combination, but I'm this is sort of beyond the realm of my understanding of, I whenever I get new drugs, there's actually a website that I can go to and it says like it cross reacts with this, and you want to make sure, but usually I think it would have been done, but at the same time I can't speak knowledgeably about it at this moment in time.
Gotcha. That's really, really cool tech. Another question from chat is, this might be in the minutia, but it's the proprietary model. I would imagine it'd be some kind of existing model that was just trained on data. But if not, can you give us any idea of the model itself without spilling too many beans?
Yeah, so I think he published a paper on this. So one of the things I can do is let me pull up, and he published a paper on the model, and it's taken a few iterations. So I can only give you like the earliest iteration that's been out in the world. And so actually, let me put out, this is the most recent one that's out. But he also used this technology to identify drugs that could be used to treat patients with COVID. So this was pre-vaccine or yeah.
That's
So
really, really cool.
pre-vaccine and being, or even for people who don't want to take the vaccine, running the test to figure out are there treatments that we can possibly give them. And so he used this to really figure out the right treatment, and they're running a clinical trial on that as well.
Gotcha. So the algorithm has this cross application of it use as well.
Correct.
So that's really cool that it was also being used for like the COVID studies as potential treatments. Yeah. Is it possible, from a question from Better Yet Shark, for a tumor that has become treatment resistant to one drug to become unresistant to it later on? Would this allow us to see if a drug is worth trying a second time?
Yeah, that's a brilliant question. And so what he's referring to, so for in a lot of cancers, when you apply a drug, it'll kill a large percentage of the cancer cells, but sometimes these cancer cells acquire a resistance and then go on. And he's asking, if you go to second line therapy, would it change around and then become sort of susceptible to that initial drug? I'm not sure we've whether that's been done, but I don't see why that wouldn't be the case, right? Like, so what I can say is that with prostate cancer, say, prostate cancer is driven by hormones, so hormones called androgens, and there's really some interesting research that it is one of the standard lines of treatment is sort of androgen deprivation therapy, so reducing the amount of androgen that is given to prostate cancer cells, so suppressing that growth factor. And but at the same time, other research from people across the world and at Fred Hutch find that like even applying a giant, huge overdose of androgen can also deactivate prostate cancer. So it's almost like a Goldilocks effect. There's a right zone that it survives, and so it's possible that there are treatments where people go low, kill off a whole set, then go really high, and then go back to low, and so switching back and forth. And this has been done for prostate cancer at the very least
And
that I know of.
do we know if that like high and low kills off different populations of cells then with like different susceptibilities, or is it just random?
It's fundamentally possible. We haven't actually done. I don't think we've actually done that sort of digging into exactly why, but we have people that are currently working on it. So actually, I believe it or not, I have a handy dandy story at hand that I can put in the chat as well for that.
Yeah, could you tell us about the story as well?
Yeah, so it is in prostate cancer exactly so basically what I was telling you about. So going in and figuring out which mechanisms are triggered by both the deprivation and which ones are triggered by the overuse to find the right exact treatment. My nose here.
I like these science spotlights, by the way. Those are really cool that y'all put those out.
Yeah, I mean, it's a really great program at Fred Hutch where we encourage. So there, it's open to trainees who want to learn and sort of stretch their muscles and try something new with regards to science communication, and so they get to work with some of the faculty and learn how to write these pieces. So yeah.
Yeah, I like there's like a nice summary figures as well of like how these potential like the concept behind the story works too. So and they look really pretty and looks like it'll be accessible as well to be able to read one of these. Thank you for sharing these with us.
Of course, and again, some of them are just a little bit more complicated than others, and that happens, right? Like science is hard, and and sort of yeah.
And then distilling it down too, because some of them it's like so deep rooted in the field that it's just hard to kind of disentangle it from like the rest of it without 20 years of background knowledge. There's at a certain point there's only so much you can do on that front, right?
Absolutely, and I wonder, I mean, if I were to have a hypothesis as to why those two mechanisms, the difference between the deprivation versus the overuse, like I'm assuming like the deprivation is not providing the growth factor, and so the cancer is lacking in like growing and being able to like the signal isn't there for it to keep growing, versus the super high is sort of flooding the available growth factors, and the growth factor, the receptor on the surface may be dimerized or homodimerized or in close proximity to other important receptors on the surface, and when you flood it, you sort of inhibit other signals coming into those cells, and so it's not getting other growth signals for it to like grow or divide or something else. And so one is like shutting down the growth signal while the other is depriving other exogenous signals coming into the cell.
Nice. So yeah, it's definitely a Goldilocks effect to it, which makes it even more complicated. You'd think you just overload it and that would kill it, but it's really interesting that it goes the opposite direction as well with like the too little of it.
Right.
And Kai, thank you for that gift sub to Better Yet Shark. Shark, enjoy that ad-free viewing now, and feel free to ask everyone, Dr. Guppy, your questions. One thing that you told us last week that stuck with us the rest of the week that I was wondering if you could retell the story, because there was so many people who were like calling back to it the rest of the week, was the connection between tooth health and cancer.
Oh yeah.
Would you mind just like retelling it to the party people, or if we have I don't know how much you can tell us about it, but if you can tell us a little bit more about it, because people were just so excited about that story.
Yeah, and there's an element of this story that actually also ties in with what soon to be Dr. Elizabeth Bonner was. I'll keep calling her that. If she's listening, she can take it up with me tomorrow or something. But yeah, it gets to that as well. So, first, with regards to Dr. Bonner's work, right? Like she's talking about recreating models in mice or near human-like, and the hard part is that um, mice are not humans, believe it or not. And as well as we can try and mimic, there are elements that are missing, which is why a lot of drugs don't make it from like that final phase to that from preclinical models to clinical tests, because there's something missing or there's an added something in the human cells. And so um, working with a head and neck researcher, first really quickly, the aside is that one of our groups basically went to a head and neck researcher and said, hey, can we get some healthy tissue from the mouth and some cancerous tissue from the mouth? And they're like, none of you have healthy tissue in our, like, humans have no healthy tissue in our mouths. Like, it's all either inflamed or like cancerous. There's no really healthy stuff in there. And so looking at those two different cell populations, one group was able to identify a class of immune cells that they've never actually seen in mice. It doesn't exist in mice. But this class of immune cells, they call them super suppressors, and they're able to shut down the immune system in and around them. And they were present in tumor cells, but not in the inflamed cells. So a certain immune cell population grows up in your mouth, and then when it gets there, then it can just like shut down the rest of the immune system, and then anything that's precancerous will grow up. So that's one part of the story about how like humans are different. We've never seen that population in mice or any other species.
What's the benefit of that cell type?
So I think the benefit of that cell type is it probably prevents autoimmune disease, right? Like it probably reduces your immune cell, like really just attacking you and like even when it's not sure, instead of like attacking you or attacking, it probably just like shuts it down.
So is it present in the whole body, or is it primarily just like in your mouth for that?
So it's a good question. They're finding it associated with an increasing number of certain solid tumors. And as Better Yet Shark says, these are regulatory T cells.
Okay.
These are the Tregs. This is one subspecies of Treg, which has certain markers on its surface that it's able to like shut down these other subtypes in there. And so similarly, working with that head and neck doctor, somebody worked with them to get like oral tissue, but also gut tissue, and looked at the association, look for the presence of certain bacterial species in the tumors for oral tissue and gut tissue. And they found this a couple of these bacteria, I think they're the treponema and Fusobacterium nucleatum. Fusobacterium nucleatum, right? That's the right word. And they're both present in oral niches and in gut niches. And so usually they're oral bacteria, and with poor dental health, they can also go to the gut. And they're really strongly associated with increased risk of aggressive cancers, both like head and neck and colorectal, to the point where it is, you can see it causing a change in the tumor cell, that it's more likely to bleb off and come off from the primary tumor and go somewhere else. And when it goes, those metastasizing tumors often take the bacteria with them. And somehow it's also able to like silence parts of the immune system, whether it's by bringing other immune cells around and sort of protecting itself or going somewhere else. So.
And it's wild, so that poor dental health will change these population numbers of the cells, and then they might migrate to the gut and induce there to be some flavor of gut cancer.
Yep.
Like, that's incredible that something like that happens. And we were like, the chat was talking about that for the whole rest of the week, because we were talking about there was some like gut microbiome relationship between different papers, and so it was just like that is really really remarkable that something like that exists and that was discovered, and it was really exciting about it. Yeah. Pragmatic entropy. You need to please DM mods and not make public comments like that. Thank you, pragmatic. It's more a businessy question from Handmade. Is there a reason to keep the model proprietary in the field?
That's a good question. I don't know whether it is proprietary in the field, and so this is one of those where I will say maybe next time I'll come back with an answer. I can dig into whether it's open source or not, but yeah.
Yeah, it's a difficult one, right? Because they also, I mean, not based on this particular lab, but there's other instances where you want to make sure that you're making it proprietary because it's also going to be funding future research as well. So it's always that balancing act of whether or not it would be, it can be, and how to be publicly available and when to be. I know there's, whenever we're making certain drugs in the lab, there's already that initial likelihood of like, how can we patent this to further benefit and use those funds, because so many drugs don't get to certain clinical trials and just fall apart and will not be allowed to further study. So I think it's a very important question, but it's probably going to be on that case by case basis.
Absolutely, and I think it's possibly just like proprietary just to make sure that they're able to get the grants, or just in case it needs to be, before like releasing it into the public. But I mean, this Taran collaborates with so many labs at the center. Like I know he's working with both liver researchers and brain researchers getting tumor tissue, and I think that's I just, for so many cancers where there's not a ton of funding and there's not a ton of research to identify new drugs, what he has is going to be life-saving, because they're just repurposing what's already out in the world, and finding some sort of treatment,
which is really cool. And both from Elizabeth and in the machine learning lab as well, there's the reutilizing of already approved drugs for new things, and I think it's just it's a really clever way of going around and trying to help people in a faster but safe way.
Exactly. You want to, because I know it's possible that the chat doesn't recognize like how long it takes to get drugs approved, and so with any new drug, like there's many phases of a clinical trial. Like there's a phase 1 where you check for safety and efficacy, and you sort of up the dosage of a certain drug, and you see whether people have any adverse effects or adverse reactions to that. And that's a long process.
Would you mind, Dr. Guppy, taking us through that process like in depth? Because
sure.
Ham says I'm not sure of the process. I'm sure there's other folks as well, or maybe watching later on the VOD, of just like, where do you start? How do you take it through the process? Like how can the process fail at each one of these steps? And in the context too of how much money like goes into that too, of like each one of these steps, if you wouldn't mind telling us about that.
Sure, absolutely. And there is also that element of like, I will say that it's really hard for a cancer center or any small group to actually be able to do all 3 phases of the trial, just because it gets so expensive to actually run such a trial, because you're basically paying for drug, you're paying for patient care, you're paying for associated treatments, you're paying for like stays at a hospital, all of those elements, and that just gets really expensive for any drug to run a trial. So the simplistically, there's 4 phases to a drug trial. There's phase 1, which is looking at safety, and so often what happens is they take a small group, a small cohort, and they give them in different concentrations or increasing concentrations of the drug to try and get to are there any side effects, are there negative effects, and you're looking across the body at different parts, and with a certain class of drugs you should be looking at certain organs and certain things more carefully. So that's the safety phase.
College students, right, or like the young individuals that are available.
So actually, yeah. So we can talk about that part in a bit, but not necessarily.
Okay.
But at the same time, it is a small cohort, and who gets offered a clinical trial is a whole different big issue that we should absolutely touch on as part of this discussion. So there's a small group. Then you move to a phase two where you up the amount of where you once you've identified the right dosage, then you apply it to a small group to look for efficacy. So how effective is it at treating the disease? And um, there's a difference between a drug that is treating a condition versus one that is protective, like a vaccine. Like for vaccines or other things that you're giving that are not treating a condition, the hard part is you have to really go above and beyond to prove that it won't have any negative effects. Versus if you're giving a treatment for a drug, you want to make sure that it is improving on current standard of care. And so you want to be able to compare it to that. Like it has a better outcome, it has better things and markers of outcomes that suggest that it has an improvement on current care, because you don't want to put people through it without that. And then you go to this phase three, which is really large. You go you go across the country, multi-center or multinational, to get like hundreds or thousands of people on the trial to give them this drug versus a control. And sometimes it'll just be like standard of care or standard of care plus this new drug, just to make sure that
So the
people
control would
have
be what we know works, what we know.
correct.
You're never putting the patients in a situation where you have this disease state and we're giving you a placebo, for example.
Yeah.
So that the disease still isn't going to progress, but it'll either be standard
Correct.
of care, or this something where in at least
Correct.
phase two we've identified it's at least as good, if not better.
Correct.
Okay.
And then you do that mega trial, and then you see if it works. And then you go, once it's approved, you have to go through all the regulations and you get it approved by the FDA. Then there's a phase four, which is continual monitoring of a drug that once it's out in the wide population, beyond the thousands, into the millions, that you're not starting to see these like side effects that are affecting a subpopulation that wasn't in the test group beforehand.
And that could be a point that the drug fails as well.
So that's once the drug is approved. This would be where you would pull it off the shelves and say, it's not working now.
Because of the population being expanded to far too,
Correct.
Yep.
Correct.
There's a lot of complexities here and a lot of nuance to
Correct.
be able to, yeah. So that's
Correct.
Go ahead.
Including what you were talking about of like who is offered a trial and not.
Yeah.
And so one of the hard parts is that we often find that the people who are currently in trials are overwhelmingly white and overwhelmingly, well, I guess depending on the treatment, they're male, female, but often male. And it's because it's in the vested interests of companies for the trials to work. Sometimes they'll create a whole lot of exclusions, and you'd be looking at people that are quote unquote termed cancer Olympians. So people who have cancer, but everything else about them is perfectly healthy, because they really want that drug to work. And so they don't want people who are slightly overweight like myself, or they don't want somebody with another pre-existing condition, or they don't want somebody who's pre-diabetic, because they feel like that will affect the outcome of the drug. And additionally, like there's the belief that people of color will not take up a clinical trial. So a lot of doctors don't offer people of color a clinical trial. And so that keeps them out of these trials. And even if they're offered, it's not like fully communicated how it would benefit them, or that it's of no additional cost. And so we're sort of keeping out a large percentage of the population that is really important to know about the drug as well.
And that can have a lot of downstream ramifications, like you were saying too. Of like it's almost like you might be building up the drug to fail by only targeting
Yep.
a certain very small subpopulation, because eventually the drug is going to go to everyone, right? So if it doesn't work in most people, then almost what's the point, right?
Yeah.
And we've got a raid in, Artology Inc. Welcome to Hack In Raiders. Those of you who don't know, Artology Inc. is an amazing artiste extraordinaire. How was your stream, Artology? Welcome to Hack In, folks. We've got a very special stream today. We actually had soon to be Dr. Elizabeth chatting with us, and now we've got Dr. Guppy from the Fred Hutch Cancer Center. We're doing a special charity stream raising money for Fred Hutch Cancer Center, where all donations are going straight to the researchers at Fred Hutch. This is part 2, Electric Boogaloo, potentially, of the stream. We had one last Monday. Our goal across the 2 streams was $1,000, and we are now in a stretch goal of trying to hit 2K. Anyone who donates $25 or more will be entered for a raffle at the end of the night to win either a Fred Hutch beaker or T-shirt. And you don't have to be here for that. You just have to have donated using the Tiltify link pinned to the top of the chat. So Raiders, welcome the heck in. Artology, how was the stream? Tell us all about it. We will show off your artwork after Dr. Guppy leaves. He's got a hard out because he's got a kiddo. He's got to have dinner and schedule, and I know all about kids and schedules. So Raiders, y'all, welcome the heck in. Sit back and relax and feel free to ask any and all questions that y'all would like from Dr. Guppy. Currently, we're talking about the drug approval process for a new drug or a cancer therapeutic and the difficulties and challenges of clinical trials and where populations are coming from. But yeah, feel free to ask any questions y'all would like. Dr. Guppy's I would say one of the face leaders of Fred Hutch. He's in all the labs knowing what's going on and making the information accessible to everyone. So he's on the front lines of science communication at the Fred Hutch, and he's an absolute legend to have on with us, and big thanks to Fred Hutch for partnering with us in this amazing opportunity.
No, I mean, thank you for having us, and it's amazing that to see this community, to see the passion, and see what you and Lita are building. Like, it's just amazing. Like, to have a science-focused Twitch, like, I could only dream of something like this.
It's it's our dream coming true as well. Shark asked a big picture question with immunotherapies, targeted therapies, and our understanding of the immune system. Is cancer treatment looking more optimistic in the coming years?
Yeah, that's a phenomenal question. And immunotherapy actually ties back to bone marrow transplant, which was first done at Fred Hutch. So bone marrow transplant was the curative therapy for many blood cancers that exist. And at Fred Hutch, we created the protocol for bone marrow transplant. And when our researchers dived in a little bit further into
and another raid. Wow. Volcano Doc, welcome on in with the massive raid. Volcano Doc, hello, Raiders. Welcome the heck in. How was your stream, Volcano Doc? Volcano Doc is another science streamer here on Twitch. Professor of volcanology, all things science. Volcano Doc, how was your stream? Welcome the heck in. Geo Jim, Volcano Doc. We have Alabama, hello. Mergy Schmergy, welcome the heck in with the Fisher raid. Hogan, hello. Raiders, great to see y'all. Today, we're having a very special stream. We have got Dr. Guppy on. We are doing a live charity stream for Fred Hutch Cancer Center, and we have research scientists on who work at the Fred Hutch, chatting about the science behind what they're able to do, chatting about the cancer treatments that they're working on. We actually had slides up from different cancerous cells that were treatments that Dr. Elizabeth, who was on earlier tonight, is working on. So y'all feel free to ask any and all questions. Guys, let's get a shout out for the lovely Volcano Doc. Again, Professor of Volcanology. There we go. Oh, and already done that.
And another shout out to, just as a personal connection to volcanology. So my wife was in a PhD program for volcanology, and she was studying the mid-ocean ridge of the Pacific, and that was the interest for her thesis project. So all the volcanologists here, I hear you, I feel you. I was living that life for a good long while, hearing the research that was coming back from my wife when they were going down into the ridge and pulling out rocks and then sampling them. So
nice. Volcano Doc said her stream was good. It was got this thing that I said yesterday wasn't going to erupt, and then it erupted today. Nice, Volcano Doc. That's then it was a good stream to chat about it. For the Raiders coming on in, let me give y'all a quick introduction. My name is Balint. I'm 1/3 of SciAnts stream. Other 2/3 are my wife Lita and our daughter baby Alona. We're both research scientists with our PhDs in molecular biology, and we are here on Twitch trying to bring science to everyone. We do microscopy, which you can see here, of cancer cells, science art, deep dives to scientific topics, science news of the week, IRL streaming where we do fossil hunts, model building, dissecting the science behind video games, and more. So Raiders, welcome in. Again, we're raising money today for Fred Hutch Cancer Center. Link is pinned to the top of the chat. We both got our shirts on promoting Fred Hutch. Any dono of $25 or more gets you in the hopper for a chance to win a beaker, a Fred Hutch beaker, or a Fred Hutch t-shirt. You too can sport these really cool shirts. It's again pinned to the top of the chat. And our guest today is Dr. Guppy. We had earlier on soon-to-be Dr. Elizabeth chatting to us about these blood cells that are cancers, in particular bone cancer. Right before the raid, we were talking about the difficulty of making drugs for drug trials and all the places where these things can fail. And now we're talking about immunotherapy right before y'all came in.
Correct.
So if y'all have any questions, please ask. We have Dr. Guppy on for about another 40 minutes, as he does have a little one, but we'll be on later afterwards looking at these cells and talking a little bit more about the biology behind them. So please, y'all, feel free to ask all the questions.
Totally. And yeah, absolutely. Thank you everybody from Volcanology from coming over. This is awesome, to get so many raiders, and thank you again to the SciAnts community for being so welcoming and great to chat science with. So immunotherapy, so what I was saying is that Fred Hutchinson Cancer Center is well known for being the place that pioneered the bone marrow transplant, and that was the real origin of immunotherapy, because once the researchers dived in deeper to see which part of the transplant was having that anti-cancer effect, they discovered that it was the T cells that went in. So a specific subgroup of immune cells called the T cells that then went and destroyed the cancers. And so they asked, could we reprogram T cells to be able to recognize all cancers that it doesn't? And there's two different approaches that have taken root. One involves basically, it's called one involves sort of taking the T cells. Both of them involve taking T cells out of the patient's body. One involves putting in these natural T cell receptors that they can figure out will bind to the surface of a cancer cell that is presenting a modified protein that is unique to that cancer cell. Another version is basically creating a Frankenstein monster T cell, which is a little bit of it takes a bit of an antibody and attaches it onto the T cell, that any cancer cell that has on its surface a protein that is not present in our healthy cells, it can now be pulled towards that, and once it can grab onto that, it will destroy it. So I think of it in two ways. One set, the one where you put in the normal T cell receptors, is giving a new prescription to your T cells. It can now see differently within the cancer cells and then destroy them. The other one I almost think of is attaching magnets or Velcro to the T cell and hoping that it sticks to that other Velcro, in the knowledge that it sticks to a different type of Velcro, and when it does, it'll stick and it'll destroy whatever's there.
And so, but that gets complicated with this idea of T cell exhaustion as well, right? Where the T cell is not functioning. I know we touched on that a smidge last week, but could you give us a little bit of an insight into what T cell exhaustion is and how that affects cancer and what work it is being done to try to combat that?
Yeah, and so I'll say two things, right? The T cell immunotherapy is so promising because it's been so curative for so many blood cancers. The first T cell immunotherapy was approved, I think, only a couple of years ago for which targeted a protein called CD19 on the surface of many blood cancers. With solid tumors, it's been just that little bit trickier because something about the environment around a solid tumor can shut down the immune system. And it can shut down the immune system in a couple of different ways. One is it can engage with the T cells on the surface and basically tell the T cells, you're exhausted, go to sleep. And so convincing them that they're exhausted and they can no longer attack. There's a couple of other mechanisms, but T cell exhaustion has been one of the the things that we've been seeing a lot. And so there's a class of drugs called immune checkpoint inhibitors, or ICIs, that exists, something like a Keytruda, where all it does, it sort of blocks the cancer cell from effectively immobilizing T cells or convincing them that they're exhausted. And so one of the treatments that we're now doing with our immunotherapies is combining T cell immunotherapy with that checkpoint inhibitor to keep the adapted T cells alive and well and healthy and attacking.
Nice. So you can counteract their shutting down almost by keeping them.
Right.
Like, it's okay to keep active, and by treating them.
Correct.
And then it's the balance of not having them overactivated, right? Like where they're
Correct.
not going to be.
That's the other part of it as well. Yeah, absolutely. We have researchers that are doing work into finding out like what are the toxicities associated with immunotherapy and how can we downregulate those. Like, because when T cells show up in clumps and start attacking, they can set off something called the cytokine storm. So these are molecules that trigger just general immune reaction and just crazy amounts of inflammation. How do you prevent that from happening? So we have researchers that are working on that as well. Right, and I saw a comment from Holy Lifton that it is not a generic treatment, and it is really tailored for each patient depending on what kind of cancer they have. It's whether you have that sort of molecule present in your tumor, then we can adapt the T cells to be able to see that marker.
And so how does one go about tailoring one of these treatments for a particular patient? Like, what does one have to do? What are the steps I mean, I presume it's going to be different for every treatment and tumor, but what are just some generic things that one does to make sure something is tailored and best outcome for a particular patient?
Absolutely. So if we're going to stick with the immunotherapy realm, what you want to do is you want to look for aberrant proteins. So proteins that are present in tumor cells but are not present in healthy cells. So there are certain times, like a protein which is only turned on during like embryo development and is shut down for the rest of our lives, gets turned on in a cancer cell. So you're safe to target them because they're not turned on in a healthy human body. And so figuring out what's turned on and is present in the cancer but not present in the healthy cells. And sometimes it's what's turned on, and sometimes it's a little bit what soon to be Dr. Elizabeth Bonner was talking about, where the process of RNA splicing is messed up and certain cells are now making new proteins that don't exist anywhere else. It's just like it is unique proteins that have unique structures that do not exist anywhere else, and all of these have to be served up to the immune cells for checking. The next phase is to see whether they're present only inside the cell or whether they're expressed on the surface. And if they're expressed on the surface, then it's a little bit easier because then you can just figure out what's out on the surface. Let's find a little piece, let's create an antibody against it, and then when we do, we can attach it to a T cell and attack. If it's adjusted inside, that's a little bit more complex, because then we start getting into the process by which proteins are broken down in each cell and presented to the immune system, and that's different, and it's connected to something called an HLA, and that's different for different people and different ethnicities, and so it gets a little bit tricky there because you have to look for the right HLA and the right like fragment of protein combined. But it's possible. We have research that is looking for those combinations of things, and then you create T cells from that, or look in the body for other T cells that already recognize that, and then copy the code and put it into a new T cell. And so we pull it out of the patient's body, we expand it in a lab, we use a CRISPR to change the T cells, and then we put it back into the patient.
It's really beautiful science to be able to do that, and just also shows how CRISPR has not only changed the world in terms of gene discovery, but also therapeutics and treatment.
Yeah, and I think eventually, like, some of our researchers are working on some kind of delivery mechanism where we can deliver CRISPR or any other gene editing software to the T cells within the body, because one of the real drawbacks to accessibility and cost are the fact that there's not that many facilities where you can pull out a patient's blood in a sterile manner, separate out the different types of cells, grow the T cells for a few weeks in a sterile manner without any sort of infection, alter it either using a delivery virus or using CRISPR, regrow it, and then put it back. Because, and again, we talked a little bit about this at the last stream. Like, whether you live or die due to cancer should not be due to where you live. This should be accessible everywhere, right? And so, if we can create the technology where we can put in the gene editing and the right things that would direct it to the T cells in your body and edit them within your body, then we could really make T cell immunotherapy accessible to way more people.
Yeah, that was a really important thing we were talking about last week too, was that accessibility component of it, which I think goes at least under highlighted in your everyday outreach, right? It's like we're talking about the direct science, but it also has to be able to be accessible to more and more people. And it's not just like at a big hospital centers, but like in rural parts of the country and world. And so it's an important thing to take into account for.
And it sort of accounts for the cost of immunotherapy as well, because right now, because of the difficulty of creating the immunotherapy, that first FDA approved one is going for in the range of $400,000 for an infusion, which is just not accessible to most people.
Right. I mean, even most of us with regular insurance is not going to cover that,
right.
So it's a doozy for sure. With respect to T cell therapy, Smikes was wondering, for that kind of therapy, does it just convince the T cell that it is out of ATC, or what exactly is the mechanism by which it works?
So, Smikes, can you reframe the question in the chat? Are you talking about the immune checkpoint inhibitor, or are you talking about the T cell reprogramming to see the T cells?
Yeah, we'll wait for Smikes to answer that. And yeah, Shark says, my jaw dropped when we saw how much it was per dose for that 400K. Yeah. I think we were actually dissecting that as Science News of the Week a few weeks ago. How does it make the T cells think it's out of energy?
Yeah. So all cells in our body have surface receptors, and the T cell has one, which is called, it has a few, but one of them is called PD1. I forget whether the T cell has PD1 or PDL1, but the tumor has PD1, and it's a mechanism that's already built in our body that basically PD1 connects with PDL1. So I think at enough use, T cells will probably produce PD1 or PDL1, and the tumor will connect with it, and as soon as it does, it shuts down. That's a signal. It's basically the body giving feedback to the T cell that you have too many T cells in the area, and you should, take a break, take a minute and go down. And so it triggers a signaling pathway that basically tells the T cell that it's exhausted.
Smikes, let us know if that answers your question. I think one component of last week...
It is biohacking, correct, Holy Lifton.
One, I think, really exciting part when we were chatting last week was just when you were telling us about news stories, unique news stories that were coming out, or research questions at the Hutch, and that was when we got into the connection between the mouth and the gut. Are there some that you can share with us tonight as well?
Totally. I mean, and keeping with the theme of RNA splicing, there's, I'll stick in that same field, right? So soon-to-be Dr. Elizabeth Bonner, tonight was talking a little bit about this process called RNA splicing. So DNA is the information that's present in every single one of our cells, but every single cell doesn't need every element of our DNA always. So, because it, as using her analogy, it has instructions on how to make a car and how to make a cheese souffle. And some cells you don't need the car, and some cells you need the cheese souffle or a hammer or whatever. And so it pulls out those relevant pieces for each of those cells. And even within those instructions, there's little bits and pieces that are not entirely always needed, and like use a wooden use a wooden spoon to stir. But if you take out use and stir, you just like add a wooden spoon to your thing, and you'll end up with a cheese souffle with a wooden spoon. So there are enzymes and there are RNA splicing elements that make a cut in one place and then make a cut in another and loop out certain middle sections to
And,
take out fragments that you don't need. Yeah,
and just
go
to
ahead.
be clear, this isn't RNase that's doing it. So we
It's
talked
not
about
RNase
yeah,
because
we talked about that on stream before, and that just chews up everything that's RNA. So these are very
correct.
particular enzymes that have very particular cut sites.
Yes.
Okay.
So it'll splice and pull out random the right elements so that you know and the beauty of RNA splicing and why it's so important to humans, right? Like the way somebody explained it to me, like yeast cells have about 6,000 genes. Humans only have about 250,000 genes. A human is not 40 yeast cells. There's something else going on in there that we're just not like 40 times a yeast cell. And so RNA splicing is the way that that's done, because what happens is you'll have this big old protein, and sometimes it's spliced one way and it'll perform one function. Sometimes it's spliced a different way and it'll perform a different function with different sort of regulatory elements. And so in some phases of its life, it will be spliced one way and be required to be shut down by this other one class of things. In another part of its life, it'll be spliced a different way and it'll be shut down by a whole another class of things. And so if splicing goes wrong, and when you're supposed to make this longer piece, you make this shorter piece, and it's no longer being regulated, that can lead to unregulated growth or unregulated elements happening. So I almost think of like the mRNA that is made into protein, like a Lego, a Lego 3-in-1 model set. Like you could make three different. Like, my daughter just got, like, it was her birthday and she got a 3-in-1 or a 4-in-1 Lego set to make unicorns and other things. So yeah.
I like the 3-in-1 analogy. That's a good one. And Holy Lifton, thank you for the 5 tier 1 gift subs to Rafiki, J Rascal, Space Stations Slovakia, the Nick Neagle, and the Wise Carrot. Thank you, Holy Lifton, for those 5 tier 1 gift subs. Also, just a reminder, folks, we have the dono link to Fred Hutch up a pin to the top of the chat. If anyone would like to contribute $25 or more, it gets you in the hopper for a chance to win a beaker and a t-shirt at the end of the night, which you don't have to be here for to claim. We'll just put your name in the hat and then raffle it off. And if we get to 2K, we'll raffle off a piece of 30 million year old amber with an insect in it. Yeah,
wow,
folks,
that's amazing.
We love those Lego sets with the 3-in-1.
I mean, who doesn't, right? But so with a lot of cancers, certain splicing factors are mutated, and like Elizabeth was saying, like it could affect a whole host of different proteins in your body. So another lab at Fred Hutch basically went about following one RNA splicing factor. So this is the lab of Dr. Rob Bradley. So they went through and looked at one splicing factor, and they're like, okay, where are you missplicing? And what types of things are you missplicing? And they found a very specific sequence that was now being spliced incorrectly in these mutated splicing factor cancers, but not in the healthy ones. And so what they decided to do is they actually created effectively a poison pill for these cancer cells. And so they sent in these little fragments of DNA with an altered splicing factor built into it that only the mutated cancers would splice. And when they did that, they would become susceptible to this drug.
Wow. So then the healthy ones are fine, right? Because their spliceosomes aren't going to target that mutated
exactly.
one. So you're only targeting the cancer cells.
Correct. We're using the cancer's vulnerabilities against it.
Wow. How far have they taken that testing? Does it works in a dish? Does it work in a tumor microenvironment? Like, does the drug get delivered into the heart of the tumor? Because like right, there's a lot of issues there with delivery to like a very packed together tumor. How good is it?
Yeah. So I mean, they've done it in a dish. They're moving on to that next phase of seeing, can we do it in a model? Can we do it in a system and be able to deliver this effectively to the cell, and in a way that the cell doesn't see a random piece of DNA and then is like, we're being attacked by a virus or a bacteria, which also fair. It's the job of the cell to do that. But then is it then converted? And they're working on that. Like, what's the best way to deliver this? Probably in collaboration with others, and on how to make this a treatment.
That is really cool. So it is dependent on a viral vector delivering this element to a group of cells, right? And that's going to be injecting into both healthy and non-healthy cells. So you probably have to figure out how to get it to the tumor site, and what.
You don't have to, right? Because again, the healthy cells would not missplice this one, so it doesn't matter where it ends up. But you got to make sure that it ends up.
Gotcha. Potentially basic question, says Smikes. I don't want to derail, but cancer is out of control cell growth. I don't want to derail. Cancer is out of control cell growth has to do with cells create a tumor.
I think what it's possible, Smikes. I'm gonna answer this, and you can fix it. I'm laughing at Holy Lifton's comment. I don't believe this is how I Am Legend started. I think it was just like Omega Man and I Am Legend had different viral origins on that one. But I think what happens is that every cell has a very precise mechanism of, when there's food, grow. When there's no food, stop growing. When you have space, grow. When you don't have space, stop growing. And in cancer, those like stopping mechanisms have gone away, and those cells just start growing out of control and taking over the healthy cells. And as Elizabeth mentioned earlier, at some level they start almost going backwards in time to that stem state. Like, there'll be some alterations, and when the space is too constrained and can't grow anymore, it'll alter and it'll like squeeze out of there and go somewhere else and form a tumor as well. So these cells just keep growing and make way more of itself and start taking more of the resources away from the rest of the healthy cells around them.
And I guess the follow-up question, Smike says, how do they become this mass? So how do you go from this like random assortment of mutant cells to this tumor?
Right. And so they also lose some level of spatial recognition. Like, every cell in our body knows, I have to be touching this and I have to be touching this, or I have to be connected here to this and here to this. These lose that ability and they start like growing on top of each other, and they form a mass altogether.
Are they bound in some way to one another?
They are bound in some way to one another, for sure. But you start seeing that at the edges, the cells, there's some research in a couple of our labs which is looking at what happens to the boundary cells and how are they changing from their original programming to become a different cell type, because they're meant to be constrained in one physical layer altogether, but something's happening that's changing in these that causes them to explore and maybe come off and go somewhere else to form. And there's some interconnected conversation. Go ahead. Yeah.
So the cells that are outlining the tumor, right, may be built, they have different properties than maybe the cells inside of the tumor. So there's like almost like an exploratory cell type versus the maybe the internal growing cell type. So that's, again, speaking to the difficulty of working with cancers, because all these cells are so different. Like, it's not just one population of cells, it's all these cells doing very, very different things.
Absolutely. And even within them, like, there's, once, as they're growing out of control, elements of their regulation have been turned off. And so they're always creating these new mutations and doing something else. And suddenly one might create a mutation that gives it even more of an advantage, and it'll create its own subgroup within it. And so while on the other side, a different mutation arose and that took over, and treating those two are very different. So that's why it's really hard, because at some level you want to figure out what drove it and what is driving all of them. But each of them has evolved other mechanisms along the way that is slightly different from one another.
And those subgroups are what's going to make it difficult for one treatment potentially to hit all those cell types, which is why,
Correct.
like that machine learning kinase inhibitor approach of like coming up with the best combinatorial treatment per patient is going to be so important.
Yeah. So the question is, so one cancer can cause can drive different cancer. Blah, 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 blah. Or are you talking about how various viral agents? So one cancer itself. So the mechanism of making cancer often breaks either the DNA repair pathway or the cell division. Something has to go off to allow these cells to divide so quickly. And these errors will spawn more errors and even more errors. And so you see, even within one tumor, there's various different populations of cell types that make up one tumor.
Yeah, and then they can spread on. You don't know which population is going to spread to the rest of the body if
Correct.
it does metastasize. So even if you've done a profiling of it, by the time maybe it does, those could have also changed.
Yep.
Yeah. Knowing that there
Exactly.
is from exploratory cancer cell that then spreads, is there any research on suppressing those particular ones? So like in particular suppressing the cancer cell types that are gonna spread so you keep it a benign tumor?
Yeah, so I'll tell you about two pieces of research with that. Both are from our groups researching metastasis in breast cancer. Because in breast cancer, what happens, often the tumor will metastasize and go somewhere else in the body, and it'll stay dormant for 5 years, 10 years, 20 years, 25 years, 50 years even, and then come back up. And it would have been from the same one, and it just then takes over the body and starts growing again. So it's really important to figure out how to stop that metastases. So Fred Hutch has launched a new Center for Metastatic Research Excellence, it's called MetX, to study the process of metastases. So these two researchers are studying two different things. One of them is studying like the exploratory cells in the edge, and what is the change that is leading them to sort of pull away from the rest of the mass and move. And while it does that, they've also discovered that these cells go in clumps, in these like groups of 4 or 5 cells, and that's more likely to survive than like a single cell going off by itself. Because these cells, when they go off in this clump, they form sort of an internal chat channel and sort of feed each other these molecules that promote their own growth to stay alive while they're not getting anything from the outside. And these are really low these molecules would normally not trigger growth. It wouldn't normally do anything, but because they have these like hyper constricted channels to chat, it's the only thing that works, and it keeps them alive until they can move to another place. So they're studying that mechanism of how cells move. So I'll pause there if you have a follow-up question, and I have another story about surviving at distant sites as well.
I wonder if one avenue would be to prevent them from migrating in groups of cells. If you could just have them migrate as lone cells, like you were saying, the likelihood of them surviving is much lower. That would be or I don't know if you can tell us like what the avenue is that they're trying to do of like breaking apart that traveling version of the cell.
Absolutely. So I think they're looking at like what are the molecules that are being used for signaling in between. So Elizabeth talked a little bit about these things called organoids, which is a way to really create a 3-dimensional version of a tissue. Mm-hmm. So you have a healthy and a cancerous tissue in a dish, and they're imaging within it and using like really cutting edge technology to actually see what the molecules are that are being used to promote that growth. And if you can start blocking that, then you may be able to prevent just those clusters from living while they're moving around.
So you can simulate a cluster in the dish, mm-hmm. right? Because I know we've talked about organoids in the past, is one of the difficulties is that it's not always like in the correct environment, or maybe it doesn't become all the cell types that you need for a given organ. Yep. But and correct me if I'm wrong, for one of these cell clusters from the tumor that siphons off, how much of that little group of cells is varied versus the same? Because presumably if it's coming from the outside of the tumor, I don't know if it's naive to predict if they're very different from one another or not.
Yeah, no, it's really all the tumor cell.
Okay.
So you're right. Like, it is not that cluster of other cells. It might have a bacteria hidden in there, and that's a different conversation.
Oh, wait, hold on now.
But-
Dr. Guppy, you got to tell
no,
us
no, no,
about that too.
we did talk about this earlier, right? Like the metastatic colon cancers
Okay.
with the fusobacteria, they often take the bacteria with them. But yeah, here the clusters are just all breast cells that are creating these internal chat channels.
And so when you're working with them in the dish for the organoids, you're trying to add maybe drugs or something that split them apart as they're coming off from the main body of the tumor.
Correct.
Cool.
So you can do it two ways. You can add a drug. You can dissociate the cells, put them in the dish with the right conditions to form a clump, and then add drugs to see which ones don't clump, versus recreate the clump and then add the drug to see which breaks the clump apart.
So you want to recreate the clump in one of those approaches?
Of course,
Yeah.
because you recreate the clump
Yeah.
and then you try and break it apart. You want to do both, because some people may have it already pre-clumped and clumps going out in the bloodstream, versus others that's not happened. You want to just block it right from at the source before it ever happens.
Are those generally two different drugs, or can they be the same drug?
That's a good question. Where they're looking.
Gotcha. Okay, I just didn't know if there's like a common theme to how that might work. That's a really exciting possibility of having that and targeting the tumor before it can spread. Do you by chance? I know we have that other story as well, but I'm just intrigued.
Yeah.
Do you know what are the factors that allow these tumors to stay dormant for so long, and then what actually ends up triggering them?
So that's the other story, actually. So
this wasn't a setup, Chad. This was not a setup. This was just
well done.
My curiosity driving us.
Yeah. So the other is like, how does it get to a different spot and become dormant? So with breast cancer, the most common treatments target rapidly growing cells, right? So anything that's rapidly growing, they assume is a tumor, but it's also like your hair cells and your gut cells. And so you often have people going through breast cancer treatment that have those side effects of like losing their hair or like other GI issues, right? Because this is targeting anything that's rapidly growing. But when they metastasize and go dormant, they stop rapidly growing. So something happens and they go somewhere else, and these cells enter a phase called quiescence, or sometimes called G0. In a cell cycle, you have a growth 1, S phase where you're synthesizing new DNA, growth 2 where they expand, and then M, which is mitosis, when they divide. These ones sort of come out of that loop and they don't follow that growth pattern, and they just live quietly with nothing. And so this other lab took a couple of different approaches. One was like basically recreating models of different parts of the body where we see metastases, and trying to see what the conditions were in those systems to allow the cells to become quiescent and evade treatment. The other way they did it was they looked, these don't metastasize to every part of the body. So like skeletal muscle never has breast metastasis. So they're like, what about skeletal muscle is preventing these cells from ever metastasizing there? So they're taking two approaches. What are the signals in one? And so they found that these metastases often go right by the blood, by like places where you have vasculature, so blood vessels of various kinds, and sort of connecting to the blood vessels will often help them sleep and be suppressed. And in that phase, they'll also retract any signals that would allow the immune cells to see them. So they're not rapidly dividing, and they've retracted anything for any T cells to see them. Wholly often, we recreated the living quiescent cells, and we're sort of studying what is the mechanism for them. And if we had the right technology, could we provide a drug that will force them to show their hand and be seen by the immune system, and then put in immune cells? Do they swarm? And they find that they can find them when you give a drug that allows it to be seen by the immune system.
So the idea then would be that you wouldn't have this 10-year wait of at some point, because right at current treatment is you don't have one. If it's metastasized and gone
Yeah.
somewhere, it's in this hidden state. There's no way to identify
Which
it.
we don't know. Yeah.
Right. And so the dream would be is if you give a patient this drug, and that would reveal whatever markers on the outside of the tumor cell and then be targeted by the host immune response, and hopefully the tumor be small enough where you don't need chemotherapy or anything else, but your immune system's good enough to fight it off.
Correct.
Wow, that is really cool. Do we know why?
Isn't it?
Do we know why skeletal muscle is where breast cancer?
Yeah.
doesn't
So with
metastasize?
yeah, I think that paper is published. So it's I think the feeling is that the environment around skeletal muscle is very toxic to the growth of these metastasized cells. There's enough damage to them that whatever is in and around the skeletal muscle like affects their DNA even more than it would a normal healthy cell.
Okay, is that something that would be possible to try to recreate as well? Like, is that an avenue where we can pursue, like, of the body's natural defenses against it, or is it one where it's such a microenvironment and so specific to this particular kind of tumor and environment that it's not currently on the docket for pursuing?
Fundamentally, I think it's option B, right? Like it's just the microenvironment in skeletal muscle is so harsh, but it does say one thing, that like things that are metastasizing are just fragile, and
Right.
they're already in a fragile state. So could we add something that would like really put pressure on them, and that pressure is normally not felt by healthy cells but is really toxic to those
I think it really
metastasizing cells.
it's demonstrated by, like, how you're saying with the sensitivity, that it's one cell can't just go off and do its thing. That's already
Right.
It has to be in that group that are communicating to one another to stay
Right.
alive, and then, like you said, go find a blood vessel to hang out with and get
Right.
some of that nutrients. And I think that's been really revealing, I think, tonight, about a little bit demystifying of how it's not just that metastasis is this crazy aggressive powerful thing, but it is in this very fragile state, and it has a lot of potential for us to target it and maybe come up with a therapeutic, which I think it's Coffee Rocks was asking about in the chat earlier tonight, and hopefully Coffee that brings across that answer.
Yep, and Coffee, there's the link for the metastatic cancer research center that we are launching, and it's really crazy, right? Like at some level, there are some immune cells that actually even inadvertently help metastases move through and end up right in the vasculature of lungs, if you will.
How does that happen?
So, I mean, they're always patrolling, right? Like
Mhm.
they're always meant to like check the bloodstream for foreign objects, and at some level, there's something happening where these immune cells are interacting with the pieces. These immune cells are interacting with these metastases, like pulling them through the cell walls of the lung and bringing them in and near the blood vessels, and sort of where they can then become quiescent. And so that's part of the research that's happening. We have a lung cancer researcher who is studying that effect, really, and it's really cool the way they do it. They can actually visualize single cells within the lungs of living mice with this technology.
Wow.
And it's like microscopy to 1,000, right?
That's amazing. Like how that single cell moves throughout the environment, and then you can mark it as a tumor cell or not, and seeing like how that movement happens.
And then you can mark different immune cells and see which ones it connects with and which ones it doesn't, and how like some of them are like moving it in and out of the lung tissue.
That's really, really cool. Wow, it's amazing the breadth and depth of work being done at the Hutch. That yeah, it's, and also your knowledge base too, to be able to stay up to date with all of these different things.
I mean, it doesn't make my job boring, that's for sure, right? Like, I get to see some of the coolest work that is happening at Fred Hutch, and I am privileged. I am fortunate to be where I am. and sort of see what we are doing in this space. Like it's stuff that I could only dream of, conceive of, like barely conceive of, and just seeing the breadth and seeing all of this, you just there's so much more, and that it sort of gives me hope that really we will, hopefully in my lifetime, have to go through another rebrand, and we will no longer be Fred Hutch Cancer Center. We'll be Fred Hutch Institute for Virology, and we can shut at the doors on cancer.
It's a beautiful goal, and hopefully we will get there. Especially, every donation helps us getting closer and closer to that goal. Because the science that we're chatting about is amazing, but it is not cheap. It is not cheap, to say the least. Dr. Guppy, I know you have to go in just about 5 minutes, so maybe is there anything you'd like to wrap up with?
What to wrap up with? So I'll say, you know, it's been an honor and a pleasure to be able to chat with you, Balint, and with this entire group. Like, I um, I'm really in awe of what you have built, and I'm eternally grateful for you and to Lita for being able to take science to the people. Like, that is really our next challenge, and I'm really um, grateful for this audience for wanting to engage and have that conversation. So, and to support Fred Hutch, like I could not be more grateful to all of you. So I will say that, and I'll say that, happy to stay in touch and maybe let you know of like other Fred Hutch virtual events that are coming down the pike. I think we might be having a virtual event talking about herpes sometime in February. So if people want to stay in tune, there might be a virtual event that we discuss herpes and treatments for herpes going forward as well.
Yeah, I think that would be incredible. I know we try to advertise any public virtual event that we see on our Discord, and we'd love to keep this up. And I know we've talked to Dr. in the past too about doing this maybe once a quarter, of doing like bringing y'all on and chatting about the science, and hopefully raising more money for cancer research. So thank you, sir, for the opportunity to be able to chat with your scientists, with you, and participate in something really important like cancer research.
We are, I mean, there's so much more research than I have time to talk to you about, and so many great communicators that I want to introduce you and your community to. So, like, yeah.
That just means we got to bring you back.
I mean, Jackie, if I know you're listening, we'll just have to figure out the next one.
Perfect. Well, thank you so very much, Dr. Guppy. You have a lovely dinner with the little one, and we will be in touch soon.
Yeah, it's dinner and board game. That's our routine every night. We do dinner and then we play a board game as a family. Message me for like appropriate board games for 4 to 7 year olds, because we're very much in that zone.
What game is on the docket for tonight?
It's a good question. I think it's my daughter's turn to pick, so we had to make it so that everybody picks, because otherwise we pick the same game every time. She's very much into. She just got the new Settlers of Catan Junior for her birthday, and so we play a lot of Settlers of Catan to start with.
That's awesome.
But I may have skipped ahead and started introducing her to like Ticket to Ride, and there's this really fun card game called Rat-a-tat-cat, which involves like trying to get the lowest hand and sort of cheating and sort of gambling on what's out there. So
nice.
Yeah.
Shameless plug, there is a game for, I don't know if you remember the Gargoyles TV show from Disney, but there's a new Gargoyles board game that just dropped. It's a lot of fun. I would eventually suggest that.
Is it?
Yeah.
Is it for kids?
I think it starts at age 7 is I think the recommended. So it might be a little bit out the range, but it's at Target.
No, hey, Ticket to Ride is rated 10 plus, and I'm not. I think she's got it. We can start that now. So
your daughter.
I don't think Gargoyles is out. I mean, I think it is appropriate based on all the other games that she's played, right? Like, so.
Awesome. Well, thank you so very
much
again.
All right,
for
Gargoyles.
For
thank you.
Hutch, and we'll be in touch. Thank you again.
Absolutely. Thank you, everybody, and thanks for humoring me and chatting with us and having so many questions. I really appreciate you all.
Appreciate you. Have a good night, Dr. Guppy.
Good night.
Big thank you to Dr. Guppy and soon-to-be Dr. Elizabeth for coming on with us and talking all things cancer research and more at the Fred Hutch.